Hello friends, today we will discuss computational chemistry and some fundamentals of computational chemistry. So it is the branch of chemistry that uses computer simulations to solve chemical problems, and it involves the use of theoretical methods and algorithm to understand behavior of molecule and materials. And here molecular modeling, it represents the 3D structure generation, manipulation and representation. Here we can see in image the model of a cell and picture of a cell under a microscope. So this is computerized technique based on theoretical chemistry methods and experimental data to predict molecular and biological properties. So molecular is something to do with molecules, and modeling is a simplified idealized description of a system or process, often in mathematical terms, devised to facilitate computer calculation and predictions, according to Oxford English Dictionary. Now these are the computational tools, and three most common computational tools are quantum mechanics, molecular mechanics and molecular dynamic simulations. So there are basically two types: non-empirical and empirical. Non-empirical has the quantum mechanics that deals with the quantum chemistry, and the basic principle of this quantum mechanics is to correct mathematical description of the behavior of electrons in atoms and molecules. So these calculations uh, are solved by this Schrodinger equation. Here H cap is Hamiltonian operator and psi is wave function and E is the energy. And the goal of this theory is to determine wave function for the drug molecule so that internal energy of the drug molecule can be calculated. And basically two methods are there: ab initio and semi-empirical. Ab initio methods has the long-term calculations. We can say in terms of density functional theory and Hartree-Fock theory. These theories are given by two different scientists, and both the theories are basically based on this Schrodinger equation. And one another methods are the semi-empirical methods, which has Austin model AM1 and parametric method 3 PM3. Now we will discuss molecular mechanics. So here we can say in long broad terms that finding the minimum of empirical potential energy function, energy of all atoms and molecules in the system. So this is the formula: the energy, which is sum of the bond energy, angles energy and torsion and charge and miscellaneous energies like dipole or dipole induced dipole. So these are the internal energy that we see in the calculations of molecular mechanics, and the goal of this mechanics is to find the accurate geometries and accurate energies for the different conformations of molecules. So in this figure we can clearly understand the basic principle behind the molecular mechanics. Here four atoms are attached, so there can be possibility of bending angle, then torsion angle and band stretching, and these angles and stretchings which has the internal geometry and non-coordinate interaction such as van der Waals forces, electrostatic forces. So some of these energies that are generated by these basics, which is called the potential energy of the molecule. Now we will broadly see the molecular mechanics force field. So molecular mechanics are basically has the force field method. So force field, what is force field? Is the mathematical function in which conformational energy of a system is studied. In 2008, Petridis and Smith J.S. has derived charm molecular mechanics force field for lignin. This charm stands for chemistry at Harvard, macromolecular mechanics. They have done the parameterization on the basis of reproduced quantum mechanics data of the model compounds. So these are the different components of force field: partial atomic charges, dihedral parameters, bonded parameters. So partial atomic charges are derived from the RES electrostatic potential fitting method, supplemented by the examination of methyl benzene / water interactions. Then dihedral parameters are optimized by fitting critical rotational potentials. Bonded parameters are obtained by optimizing vibrational frequencies and normal modes. The overall good agreement between the structural properties of molecular dynamic run and experiment provide confidence that force field will be successful in the simulation of lignocellulosic biomass. Now charm potential energy function of a molecule approximated by this equation. So this is the equation that was derived from the previous slide that we have discussed. So all the energies by the software is calculated by this formula, and on another side we can see the softwares available for the molecular mechanics is MM2 for small molecule, Amber, Charm and Bio. These are the software for the polymeric molecular study. Now docking calculation, we will discuss the docking is basics. We already know the computational process of searching a conformation of a ligand that is able to fit both geometrically and energetically into the binding site of the protein. So this is the ligand and this is the protein or receptor that we dock. We have to generate the grid, then we supposed to fit the molecule inside the protein or receptor. Then we will see the different types of interactions like non-bonded interactions or hydrogen bond, covalent coordination. Different bonds are studied. So in this work they have performed THPP on the surface of lignin molecule by the Autodock Vina software. So here 1000 snapshots were taken regular interval during 100 nanosecond heating simulations. THPP was then docked on the wall polymer for each these snapshot with exhaustiveness factor of 20. Now free molecular dynamic simulations. So the most confusing topic of molecular modeling is molecular dynamics. So we can see it is the form of computer simulation in which atoms and molecules are allowed to interact for a period of time. The basic goal is to find out dynamic state of molecule in 3D, three-dimensional physical movements of molecules and atoms within 1 nanosecond or 20 nanosecond. We see the dynamic state of the molecule with respect to the time interval. In this graph we can see the which is derived from the RMSD in nanometer and the time in picosecond. So Merchant et al. Has performed free molecular dynamic simulations using GROMACS 5.1. So they were integrated using 2 femtosecond time step, the leapfrog Verlet scheme. Cutoffs of Coulombic and van der Waals interactions at 1.2 nanometer was taken. Then switch function was used for van der Waals interaction between 0.9 and 1 nanometer. Other side, temperature and pressure were kept constant at 290 Kelvin and 1 atm isotropically. After an energy minimization using steepest descent algorithm, 200 picosecond equilibrium simulation was run with V-scale thermostat and Bernard Brosted with this 1 picosecond compressibility and 4.5 and 1105 bar. So dialysis molecular dynamics simulations. Here in the picture we have seen before the docking, the simulation was performed. So this was performed with by replacing the acetone solvent with water. Then QSAR. QSAR is a little bit different from the previous molecular modeling methods. So here we see the quantitative structure activity relationship for the understanding of the mechanism of chemical modification of wood by method of acetylation. So this was the previous study, and this is the basic mechanism. Here QSR correlates the physicochemical or molecular properties to biological activity in terms of an equation. So these are the physicochemical properties like lipophilicity, polarizability, electronic properties, steric parameters. In figure we can see the how the 2D structure was built in 3D model. Then 3D model to generate the pharmacophoric features. Then 3D QSR is the advanced, and the 3D QSR stands for three-dimensional quantitative structure activity relationship. Quantitative relationship between the biological activity of a set of compounds and their three-dimensional properties using statistical correlation methods. For example, these are the latest technologies of QSAR. COMFA, Dylomos, COMSIA are the other type of methods. So here we can see the place we have to place the pharmacophore into the lattice of grid points. So these are the grid points which are already inside the box. Then we have to put the position of molecule which match to the pharmacophore. So the probe atom is replaced with each grid point in turn, which measures the steric or electrostatic interaction of the probe atom with molecule at each grid point. So here we can see the different types of parameters and their descriptors. So these are the hydrophobic parameters like partition coefficient log p, hence substitution constant pi, hydrophobic fragment constant f, and distribution coefficient log d, and apparent log p, capacity factor in HPLC log k dash w, then solubility parameters, that is log s. Now electronic parameters. Electronic parameters are Hammett constant sigma, sigma plus, sigma minus, Taft induced polar constant sigma star, Swan and Lupton field parameters, ionization constant pKa, delta pKa, chemical shift of IR NMR. Then steric parameters are Taft's steric parameters, molar volume, van der Waals radius, van der Waals volume, molar refractivity MR, Peracor, sterimol. Quantum chemical descriptors are atomic net charges Q sigma, Q pi. Then super delocalizability, energy of highest occupied molecular orbital HOMO, and energy of lowest unoccupied molecular orbital E LUMO. Then special descriptor JERS descriptors, shadow shadow indices, radius of gyration, principal moment of inertia. So molecular descriptors, these are, we can define a numerical representation of chemical information encoded with a molecular structure via mathematical parameters. So they showed that electrophilic index gamma in combination with other descriptors LUMO, energy, HOMO, hardness eta, and chemical potential mu prove their utility in predicting the chemical modification of food gain in mass. So these approaches allowed us to gain this formation information like electronic descriptors, energy of HOMO, LUMO, eta, mu, and omega of reagent influencing the modification of biomass surface. Then 3D QSR model allow us to indicate the appropriate reagent for chemical modification of natural fiber. The results obtained show the cellulose and hemicellulose are sensitive to the modification of their surfaces. Okay. Thank you.